So if we look at this example, really these beginning diagrams here are what we would define as the zygote. And then we kind of cut this next section up into the embryo, and then that leaves us with the fetal period afterwards. So moving from the conceptus, which is really the merging of the male and female DNA, we then move into what we call the zygote, the embryo. And really the embryo is so important, and there's a whole discipline dedicated to this subject. So if you're really interested in reproductive physiology, this might be a great avenue for you. Embryology is really that process of studying embryos and understanding how we go from just one cell that merges, sperm and egg, creates this cell, and then it just begins to divide and create organisms. And this is always the part that just blows my mind when we're looking at embryonic development. And the way we define an embryo is an organism in the early stages of development. So it's often before we can really determine what the organism is, like we're seeing here on the right-hand side. But then we're starting to see crucial systems develop, such as the heart and the lungs. And so you'll start getting heartbeats really early in most organisms. And so once we leave this zygote, after we've seen ovulation, after we've seen fertilization, then we really get into this process of the embryo. And this is the early stage of the embryonic development when we're really starting to see cells divide and start to just duplicate and create the beginning stages and the different types of cells that are going to differentiate into different things. So early embryo development is really split, like I said, into two stages. So the first one is cleavage, the second one is differentiation, and the cleavage is really just those beginning cell divisions. This is where we're going to see mitosis kick in again. There is no growth. We're just creating cells, dividing more cells. Whereas once we get past that point, we actually get to what we call the blastocyst. Then we move into differentiation. And differentiation is going to start forming layers. We're going to start seeing cells differentiate to be certain parts of a placenta or of an organism, or starting to become different types of tissue inside the body. And so differentiation is going to start building that organism from a cellular layer. So this first stage, cleavage, really goes from, we started with one cell, which would have been our zygote in the conceptus stage, and it begins to divide. It then moves into two cells. Those two cells divide, become 4, then 8, then 16, and then 32. This is really called the morula. And these are where we start to see really early divisions. We're trying to create multicellular embryos. So this wouldn't happen in bacteria. This wouldn't happen in viruses. This wouldn't happen in protozoa. This would be in some type of plant or some type of animal. And again, we are seeing only divisions. Nothing is growing. We're not seeing that hypertrophy or cells increase in size at this point. We're just seeing cells increase in number. So as we enter cleavage at the 2-cell level, we're then, like I said, going to move 4 to 8 to 16 to 32 until we get to this beautiful structure called the blastocyst. And the blastocyst is going to be 100 or more cells. And sometimes you'll see a blastocyst with a layer on the outside of it called a zona pellucida, and sometimes you'll see a blastocyst that's hatched from that layer as it's getting ready to implant into the uterus. And a blastocyst is amazing. So this is really where we see the fertilized egg, as it's dividing, is going to move its way down through the oviduct, doing all of these divisions until we get to the endometrium layer on the uterine wall here. And this is where you're going to see that blastocyst implant itself and start to grow and create placental membranes in combination with the female. And it's amazing because it's going to free float inside the uterus for quite some time, depending on the organism. And as it's doing that, you're going to see 3 distinct layers in the blastocyst that we'll talk about in just a second. And then it's going to implant into the uterus for the rest of the gestational period. Here's some extra diagrams that I really love. So this one on the far left really shows you that we go from, again, this zygote, so this conceptus, we move all the way through the blastocyst until we hit the embryo. And that's really going to be kind of what we define as an embryonic layer, as it's going to be implanting into that uterus. There's another diagram kind of going through that here on the right-hand side. And then the actual parts of the blastocyst here on the left. So the blastocyst is made up of 3 parts. We call this the blastocele, the trophoblast, and the ICM, or the inner cell mass. And each one of these has different functions inside the organism. So the trophoblast, this outside layer here that's yellow, is really going to be what turns into the placenta as it sinks into that endometrium tissue in the female. Whereas this chunk of cells up here are what we call the ICM. And this is where the fetus starts developing. And then we have the blastocele, which is going to be this beautiful fluid-filled cavity that will be inside the embryo as it begins to develop. And once the blastocyst hatches from that outside layer, the zona pellucida, you're going to see it begin the process of implantation into the female. And that's really what we're seeing here is this transition of the early blastocyst that's hatching out of the zona pellucida layer, and then implanting, now that it's shed that layer of cells, into the female. And that really leads us into what we call differentiation, and this is still kind of part of that beginning embryonic process. And so the blastocyst is going to have this layer, the ICM, and this is really where we're going to start to see primitive organ systems developing. You're going to also hear these referred to as embryonic stem cells. So if you've heard stem cell research, that's what these cells are, is that they essentially have the ability to turn into anything. And so the ICM starts to differentiate into the endoderm, mesoderm, and ectoderm. So think endoderm, inside the body, internal organs; mesoderm, think structural, think muscle; and then ectoderm, think external tissues, things like the nervous system and skin and hair. And so they're going to start differentiating into specific animal cells inside the body. And here's some more examples if you're kind of interested in where this goes from there. There's going to be a lot more development that happens between the zygote and then when we see an embryo that we can actually recognize as more than just a pile of cells. And so as you see here, the zygote's going to actually go to a blastocyst. You're going to see the trophoblast and the inner cell mass inside of that. So the trophoblast is going to become that placenta, and the inner cell mass is going to start differentiating. And so it's going to actually start creating what we call the epiblast and the hypoblast inside of there. And those are going to each have different types of things. And that fluid-filled cavity, the blastocele, you're going to see also play a part in the development of the early embryo as we move past the cellular division into the differentiation phase. And you're going to see, again, we're going to start getting all of these specific layers. So you're going to start seeing the beginning of the amniotic sac. You're going to start seeing the epiblast and the hypoblast start to differentiate. You're going to start seeing that blastocele become what we call a primary yolk sac that feeds the offspring until they implant to the maternal resources. And then again, you're going to see more and more of these cells to start to differentiate. So if we were to take that cell and cross-section it, like they're showing you here, you would actually start to see the ectoderm and the endoderm here. You're going to see what we call the primitive streak, which is where we're going to start to see the develop of the neural system, and then the hypoblast and the yolk sac are still present. And then again, it just continues to grow in length. And so you're going to start to see there's an ectoderm here that's starting to develop. You're starting to see the aorta, you're starting to see endoderm, you're starting to see the brain. So all sorts of different things start to develop. And again, this still doesn't really look like much, but it's really that, again, differentiation of cells as an embryo is developing. And then you're going to start to see the process of the mesoderm developing and that early gut as we're starting to get more and more of those organ systems in place.